Well, tonight's speaker, Maria Castano, is a Colombian biologist and part of the lab of a past speaker that we had, Albert Louis, a few months ago. She obtained her Bachelor's of Science at the Universidad de los Andes. Is that close enough?
Yeah.
Where she had the amazing experience of performing a long-term population monitoring study. She founded her own bird watching company in Colombia called Amazona Tropical Birding, which made her a more devoted birder and gave her the possibility of traveling to remote areas of the country, always making sure that local guides and communities obtained from birding tourism an incentive to get involved in conservation. Maria joined the TropBioLab at the University of Rochester in the fall of 2020. Please welcome Maria.
Okay. Thank you all for having me. I'm very excited to be here and to share with you what I've been doing for my PhD this past 5 years since I came to Rochester. And I'm also going to talk to you a little bit about my story and how I landed where I am right now. So we're going to go through a little bit of genetics at the beginning, and then we're going to move to some exciting behavioral data that I have for this fantastic species. But first, in all my talks, I like to show this slide to bring awareness about Colombia's situation right now and what doing field work in my country implies. So I took this picture in a town in the middle of the Pacific rainforest. And it describes perfectly Colombia's most amazing wonders, but also its most horrifying truths. So we are one of the most biodiverse countries in the world, #1 country in bird species and orchid species. Half of the country is covered by the Amazon rainforest, and the other half is divided by mountains that create these amazing ecosystems. But a 60-year-long armed conflict, plus narcotraffic and other illegal armed groups are a constant threat to rural populations in the country. And so violence is really pervasive, and there's a lot of deforestation. So here you can see they're trying to represent all our diversity, and then deforestation to build the roads, and then we have like the guns and the tree cutting. And so I normally end with this slide, but today I wanted to end in a more happy note. But I just thought I'd start by showing you a little bit of where I come from and what doing fieldwork there implies. So Colombia signed a peace deal in 2016, where the biggest guerrilla armed group, so the rebels, signed an agreement to end the war, to cease fire, and turn themselves in 2016. And so they left many unexplored territories that were unreachable for scientific exploration before because they were very dangerous to go to. And so this like new door opened for scientific exploration. And so in 2018, shortly after the sign of the peace deal, and after I graduated from my undergrad, I had this amazing opportunity to join the first scientific expedition guided by ex-combatants from these guerrillas from the FARC group after they signed the peace deal. So they were previously controlling these areas, and now they were going to guide us to enter the territory and join us in this scientific exploration and collection to understand how was the biodiversity there, which species were present. So it was a really fantastic opportunity, and it sort of changed the way I view the world and my country. It was a very, very life-changing experience, taught me about forgiveness and compassion, and made me a more, I don't know, grounded person. So all the ex-combatants knew a lot about the forest, way more than probably what I could teach to them. I think I learned way more from them. And at the end, they were just people, regular people like you and me, that had their own conflicts, their own fights, their own life histories that that put them where they were, and they were just trying to get through life the best way they could. So I understood that peace had to come from each of us, each Colombian citizen, and from forgiveness. Otherwise, we were not going to move forward. And so there in the middle of the jungle without uniforms or clothes that can distinguish us, you can't tell who is an ex-combatant, who is a researcher, right? We're just Colombian people taking a step into a direction that hopefully would lead Colombia to peace. So after having that experience, I was strongly convinced that Colombians should leverage this spectacular biodiversity that we have and make ecotourism become a significant part of our economy, right? It should be a source of income for rural populations, which also would help preserve our ecosystems. And so that's why, as David said, I try to support local communities with the field work that I do, and sort of try to get them involved in conservation and try to make them understand that we can also have some gains with preserving the land, right? And so I founded a bird watching company before coming here for my PhD. We were supporting local businesses, and we were featured in a couple of really cool news pieces. We got to take some really fun people bird watching, Jennifer Ackerman, who wrote fantastic books about birds, and some of the eBird creators even went to do some bird watching with us. And then after being in this business for about 3 years, I met my advisor, Al Uwe, who gave a talk, I think last year for you. I took him bird watching around Bogota, my native city. And then 5 years later, here I am in Rochester with him as my advisor for my PhD. So as a young birder in Colombia, as I said, the country with most bird species in the world, around 2,000 species, I was always curious about why my country was so diverse and what were the biological processes acting to promote and maintain this diversity that we see. Which is why I got interested in speciation, which is when one ancestral species divides into multiple descendant lineages that undergo genetic differentiation and then eventually become reproductively isolated during the course of evolution. But how and what exactly happens at this moment where lineages go down distinct evolutionary trajectories? So how one species becomes two. So it has been proposed that the main way in which this happens is through some form of geographic isolation. So this geographic isolation can be in the form of separation across altitudinal gradients, or with a geographic barrier like a mountain in the middle, for example. And then after accumulating differences that arise during that period of separation, we can have what my lab calls the moment of truth in speciation, which is when those two incipient species come back into contact and interbreed. And so we can test sort of how good of a species they are. So this area where they interbreed are called hybrid zones. And they had been proposed to be these natural laboratories of speciation because they allow us to study the effects of interbreeding and test these reproductive barriers that should have arisen the same way as we would execute experiments in the lab. And so at hybrid zones, evolution can take different paths. So if the two incipient species interbreed before they achieve complete isolation, they can fuse back into a single population. And so alternatively, if they do reach sort of good species status, so complete reproductive isolation, they can keep separate and stay as good species. Or if complete reproductive isolation is not achieved, they can stay differentiated, but they can form these hybrid swarms in the middle. So they stay differentiated, but keep the stable hybrid zones in the middle through which they can sort of exchange adaptive traits using that hybrid zone as a conduit between the two species. And so hybrid zone dynamics can tell us a lot about the fate of those incipient species and sort of how the speciation process works. And so with that context in mind, today I'm going to talk to you about a very cool hybrid zone. I'm not biased. That is my study system for my PhD. This hybrid zone sparked my interest back in Colombia when I was guiding birding tours and enjoying life before my PhD. And so in Colombia, where the Andean Cordillera ends, it divides in three large mountain ranges: the Western, the Central, and the Eastern Andes. And they divide the country and create these two huge river valleys, the Cauca Valley to the west and the Magdalena Valley to the east. And so along the slopes of the Western Cordillera, we find our two superstars of the day, the Flame-rumped Tanager, or Ramphocelus flammigerus subspecies complex, where the two subspecies exhibit very subtle morphological differences, but as you can see, they have this very dramatic variation in the plumage color of the rump. And they vary along an altitudinal gradient. So the smaller lemon-rumped tanager inhabits the coastal plains of the Pacific from Ecuador all the way up to Panama, whereas the larger flame-rumped is an endemic to Colombia, and it's found in the Cauca River Valley at mid-elevations, so higher elevations than the lemon-rump. And so wherever the two subspecies overlap at low mountain passes in the Western Andes that act as bridges that connect the two populations, they hybridize extensively and have established at least 3 different independent points of contact where they're hybridizing. And so I always say this, and my advisor always rolls his eyes, but I always say that they are the living proof that yellow plus red equals orange. And so more extensively, more technically, extensive interbreeding between them along this altitudinal gradients results in this beautiful gradient of coloration and body mass, where hybrids are intermediate size with different tones of orange in the rump. And so in this picture, we have males on top, females in the bottom from both taxa, and then hybrids are in the middle. So you can see that they have strong sexual dichromatism, so females and males are different. They're generally socially monogamous, but likely they have some degree of polygamy because we often see them in like small groups with one male and a few females. Also, variation in the rump color is probably influenced by the environment to some extent because the carotenoid pigments that they deposit in the feathers are obtained from the diet. So they don't synthesize them de novo, they have to eat them. But the ability to process and store these carotenoids is still hereditary. So it's a trait that is potentially impacted by both environment and genetics. And so if you're wondering about habitat differences, there are many. Both species are currently very well habituated to people. They thrive in intervened ecosystems. So basically, you won't find them in pristine forest. If you go to a very preserved patch of forest, they won't be there. But you would very likely find them in bird feeders, alongside roads. They really like secondary forests. Plantain and coffee plantations are also a big favorite, but they do need some secondary growth forest nearby. And so in the past, we think that they potentially evolved along like forest and river edges, or if there were landslides and then secondary growth after those landslides, that's probably where this species evolved. However, the lemon-rumped is found in the Pacific rainforest, which is the most humid place in the planet. Whereas the flame-rumped is found higher up and to the other side of the mountain in the much, much drier Cauca Valley that is also more disturbed and has higher human presence. So there is very striking habitat differences. And so if you go to Colombia, you'll find the lemon-rumped almost anywhere where there is secondary growth along the road. Notice there's like deep fog and dense vegetation that's very much the landscape that you'll find in the Pacific rainforest. Whereas the red subspecies inhabits this never-ending lands of monocrops and plantains and coffee with a little bit of secondary growth in the side. So the habitat differences are very striking. And so for my research, I wanted to explore what is the evolutionary trajectory that these two subspecies are in. So which of these three outcomes that I talked to you about is the most likely in this hybrid zone? So did they not accumulate enough differences before hybridizing? So are they going to fuse back into a single population, or are they on the way of becoming two full good species? Okay, so to answer this question, my research explores several different biological scales using an integrative approach that goes from characterizing how much genetic differentiation there is between the two subspecies, how much gene flow there is between the two, quantify individual variation in behavior and morphological traits, and measure the feather color and the pigments present in them, all the way down to the genes responsible for these amazing, beautiful colors and how they can get messed up in hybrids. So we try to encompass all of the aspects of the biology of these birds. So for the first part of my research, I wanted to broadly describe the three replicate hybrid zones that exist in the Western Andes that I talked to you about. And so specifically for this part, we wanted to know how much gene flow there is, in what direction is it going, how old are these hybrid zones, so how long ago did they start hybridizing, and in what trajectory they are. And so to answer these questions, we needed to obtain genetic data. So we went out and with a lot of effort and logistics, we sampled all three hybrid zones. So we did a geographic transect, sort of a straight line that crosses the mountain through the only three roads that connect the Pacific rainforest with the Cauca Valley. And so the sampling methodology consisted in setting up mist nets. And so the birds get tangled. I'm sure you've seen some version of this. And then first I would take blood sample from the major wing vein, and then I would proceed to band the bird. And then I'd take morphometric measurements and document body condition status like fat percentage. And so, and then I would find the bird's age based on molting patterns and plumage color. And then I would pluck some feathers too, because we were interested in doing color analysis. So here's a very fast time lapse of that process of taking the blood sample, banding the bird. I have the Ramphocelus there in my hand. And so I just measure, take the blood sample, band the bird, take the feathers, and then I do a very short photo shoot before the bird leaves. So we did this same procedure with each of our more than 200 birds that we sampled during the field. And so before I move on, I wanted to show you the results. I wanted to show you a little bit of what doing fieldwork in Colombia in these regions is like. So as I mentioned, there's a lot of internal conflict and political unrest. And despite the peace deals, there's still sort of like a harsh road ahead to deal with all the other illegal actors that patrol these areas. So the peace deal in 2016 opened the door for a lot of deforestation. So every year that I went, from when I started in 2020 up to this year, deforestation was growing at alarming rates in areas that were sort of inaccessible before because they were guarded by the rebels. So in some sense, the rebels were protecting the forest, which was a little bit, I don't know, we were torn by it. Additionally, this extremely mountainous topography makes the roads a little bit unstable and unpredictable. But I had the right connections and was taking the right precautions, so I was able to move around the territory safely. And for that, I really need to thank my amazing field assistants. These are Manuel and Santiago, without whom all of this work that I'm going to show you would have not been possible. They were really fantastic. They helped me set up the craziest mist nets, either in the middle of the jungle or in someone's front yard. Really nothing in between. These birds are extremes. And they followed me to every crazy location in very remote regions where the mobile lab was set in anywhere that we can find a flat spot on the ground. So abandoned houses in the middle of the forest or in the back of the car or wherever we saw a flame-rumped tanager. So I really can't express how crucial they were for this project. But not everything was that rough. The advantage of studying birds that are so well adapted to humans is that sometimes we have fancy birds that live around fancy reserves, and so you can have tea and breakfast while you wait for the bird to just fall in your mistnet very nicely. So it was just a very diverse and changing experience at every locality that we visited. And the perks of doing fieldwork in Colombia is that wherever you set up a mistnet, you are for sure going to find a lot of wonderful birds. So I was looking for one of the most common tanagers in the Andes and ended up with birds like this one in our mistnets, the best day ever. We caught both male and female of the Andean cock of the rock just accidentally. We were not looking for them. They just were in the mistnet. And so now I'm going to spend just a few minutes going through a few slides, showing you some of the birds that I accidentally caught while doing field work. I always want to show these slides in the talks that I give, but normally I'm talking to geneticists, so I'm not encouraged to share as many bird pictures as I would like. But I thought this was the perfect occasion to share our appreciation for these beautiful creatures. So we caught lots of different mannequins, striated mannequin, white-bearded, yellow-headed, golden-colored, a lot of diverse random birds here, barred-crested anthrax, spot-crowned barbets, hawks, puffbirds, toady flycatchers, moths, gnat wrens, a lot of Inca jays and black-chested jays, all the hummingbirds that you can think of. This one was my favorite, the white-tipped sicklebill. It was fantastic. But we have hermits, Jacobins, black-throated mangoes, violet ears, emeralds, and then my second favorite, the booted racket-tails. They are really, really wonderful birds. And then, of course, I can't leave the tanagers behind because they're just so wonderful. The tanager diversity in the Western Andes of Colombia is something really out of this world. I can't explain. This is just like scratching the surface of the amount of tanagers that we can see there. So here we have another species of Ramphocelus, the crimson-backed tanager, golden tanager, golden hooded tanager, black cap, barrel spangled, all the tanagers. I can go through all of them if you want. Bay-headed, we have the metal green, plain colored. That's really unfair that they call it plain colored, but that's fine. And then we have a few more birds, some tanager allies, honey creepers, sparrows, flower piercers, grass quits, euphonias, dacnys, brush finches, and then we also have some woodpeckers. This one you have the acorn woodpecker. It's really interesting. It extends its distribution all the way down to Colombia, and it builds granaries there, but they don't use it to store acorns because we don't have acorns. So they retain the trait of building the granaries, but they don't need it because we also don't have winter. So it's really funny to see them store rice. Yeah, I don't know. And then we have some warblers, just to feel close from home. We don't get them with the reproductive plumage in the Western Andes. So I only saw a black-burnian warbler with like full plumage when I like came to the States, which was fascinating. I also had never heard them sing. So coming here was just like, I was very disoriented. And then finally we have, last but not least, some beautiful but terrifying creatures to have in the mistnets, like this colored aracari, which you don't want to put your fingers anywhere close to that, or Craster oropendula, or some night creatures, if we left the mistnets open a bit too early or too late. We could find some band-winged nightjars around. So I diverted a little, digressed, but I wanted to show you just a little teaser of what doing fieldwork in Colombia is like and how fun it was to just set up mistnets in the Western Andes. So back to the research for the sake of time, but I'm happy to show you more bird pictures later. So we were able to sample 8 localities in each of the transect for both males and females. So maximum elevations of 2,000 meters in the higher point, and we were at sea level in the lowest point. This is just like how the transects look like. And this is the amazing, fantastic variety of rump color phenotypes of the birds that we were able to catch. So this is for the males. You can see we had like the full spectrum from yellow, a bunch of orange phenotypes, and then all the way down to pure red. And then for females, the rump varies, but you can notice that they have a chest band that also varies along that hybrid zone. So you can see that both traits are following the same pattern in females. And so to date, we have a pretty great data set for our 3 hybrid zones. So 3 transects of about 130 kilometers each, with more than 200 blood feather samples for both males and females. And so after we completed the sampling, I came here to the University of Rochester, and we did some DNA extractions and sent those for sequencing to get some genetic data to see what was going on with this hybridization. So for the color analysis, we used spectrophotometer, which you can see in the picture there. So this measures the color reflectance from the feathers. So this is an example of how that looks like. So you basically just like put the speck on top of the feather, and then it creates this curve. This is called a reflectance spectra curve. And you can see that fantastic color variation. So from this data, this reflectance data, we can calculate 22 different color variables. And so we use hue value. So the hue is the wavelength at which this curve that I show in this graph achieves the maximum slope. So yellow colors are a little bit lower in the wavelength range, and red colors are a little bit higher in that range. And that way we could assign an actual like quantifiable color to each bird, instead of me in the field being like, I think this is a hybrid, I don't know, this looks a little bit orange. It was really tricky, because depending on the light, depending on the diet, right, you can have a pure red bird that is genetically red, but then if it didn't eat or it didn't have a lot of resources in the past season, then the plumage would suffer. So it was tricky, but this way we could do a more objective measurement. And so then we looked at all of our genetic data using this principles components analysis. It sounds complicated, but basically this takes a lot of data and reduces it to essential traits for easier analysis. So imagine you have a huge collection of information with a lot of different details, like a giant spreadsheet with many columns. And so a PCA helps sort of simplify that information by finding patterns and combining similar details. So it condenses a lot of information in just a couple of few big pictures, which in this case are called principles components. So then we can understand the main trends. So we do this with the genetic data just to see what's the level of differentiation between the 2 groups. And you can see here on the x-axis that separates our yellow birds from our red birds. And that's sort of a 30% separation. So that was just to confirm that they were actually genetically differentiated, and they're not just like a single species, the same genetics, and they just have maybe a random mutation that makes some yellow and makes some red. Like they're not races, they're actually like 2 distinct lineages, right? And then the second principal components, it's just separating it by geography. So it matches really nicely. Here the points are the shape of the points corresponds to each transect. So the squares are from the south, the circles are from the middle, and the triangles are from the north. And you can see that it matches the geography really well. So the southern points are here, and then the 2 northern ones are closer together. So this was just to see the variation at the species and population level. Then when we zoom in a little bit, we can build these kinds of plots where we can quantify the variation at the individual level. So here, each bar is 1 bird, 1 individual. And the different colors show the proportion of ancestry from each parental subspecies. So basically, we're showing how much of the genome of each bird comes from lemon-rumped genes versus flame-rumped genes, right? And so this proportion we call the hybrid index, and that's represented by the letter Q throughout my presentation, but that doesn't matter. But so if you have a hybrid index of 1, it means that you're fully red. So 100% of your genes come from the flame-rumped subspecies. And you have a hybrid index of 0, it means that 100% of your genes come from the yellow subspecies, so you're pure. And then anything in the middle, even if it's a little tiny bit in those bars, means that you have some sort of admixture in your genome. So you're a hybrid. So we were able to quantify that hybrid index and then match it with the respective rump color, right? And so we did this for the 3 transects, and we found this really cool pattern where the hybrid index, here is in the y-axis, correlates almost perfectly with the plumage color. So basically how orange you are depends on how much of your genes come from the red versus the yellow. So it's like mixing paints in this case. So this pattern was really cool. So we had some idea of how differentiated the species and the populations were. And we know that there is individual variation in color that depends on the proportions of red versus yellow. But we were also interested in quantifying then how much gene flow there is, how long ago did it begin, and in which direction it's going, right? To see that evolutionary trajectory. Whoops, I didn't do that. Okay. So I'm not going to go into the details of this analysis, but we did some demographic modeling, and then we were able to infer that hybridization began in the southern transect first, around 6,000 years ago. Then it started in the middle transect around 2.5 thousand years ago. And finally, the third transect is the youngest, and hybridization began there around 1,000 years ago. So the southern hybrid zone is the oldest, the northern hybrid zone is the youngest, and that suggests that the yellow subspecies sort of was expanding to the north through the Pacific from south to north. So from this analysis, we also found that the lemon rump birds are disproportionately contributing genes into the hybrid zones in all 3 transects. And we also found that hybridization has been increasing in recent years. So we're finding evidence for an evolutionary trajectory of fusion, right? So their gene flow is increasing between them. So to complement this analysis, we wanted to obtain just a little bit more detailed resolution on how this disproportionate contribution from yellow into the hybrid zone works from a geographic perspective. So this is going to be the last genetics of this part of the talk, so bear with me. It's a very crash course on how we study this. And so to answer these kinds of questions, to get that geographic resolution, we normally use something called clines, which help us illustrate those gradual changes in the frequency of morphological traits, which in this case is plumage color. That's always going to be in the y-axis across a geographic transect. So the geography is going to be on the x-axis. That's just what I just said. And so a species can have as many clines as it has traits. So you can build one of these clines for any trait that you want. Body size, beak size, I don't know, all the things, different patches of color. And so in this example, the multiple clines belong to different traits in different colors. And so the theory says that if there's no hybridization outside of the hybrid zone, then clines should be all centered on the midpoint of the hybrid zone. But if there's gene flow from one species into the other, then the cline center can be displaced into the distribution of one of the species. So that position can tell us if gene flow is happening and in what direction. But the shape of the cline will inform us on the strength, on how strong is that reproductive isolation, because that shape is determined by a balance between dispersal from the parentals into the hybrid zone and then selection against unfit hybrids. So how bad are the hybrids produced? And so these clines are expected to be narrow if there's strong selection, or if gene flow is limited, or if there are very steep environmental gradients, and they're expected to be wider if gene flow is more extensive or environmental gradients are more gradual. So, quick recap, steep climb suggests strong reproductive isolation. A wide climb points to weak isolation, and a displaced climb suggests gene flow from one species into the other. And so basically, these climbs allow us to pinpoint traits or genes that are moving freely between taxa and how much they are moving versus those that don't transverse the hybrid zone, which are potentially involved in reproductive isolation. So we built climbs for that hybrid index that I just talked to you about. That was our genetic trait. And for the rump plumage color, which I also just talked to you about. So we just wanted to see how much the rump plumage color varies along the hybrid zone and how displaced it is from the genetic center. So these plots are going to have quite a bit of information, but I'm going to walk you through it, and then we'll move to some fun behavioral experiments. So the center of the hybrid zone is represented by the red line in the plots. The climb for the hybrid index is in black, and the climb for the rump color is in orange. So males are solid lines and females are dashed lines. And so in the oldest hybrid zone, the southern transect one, we found that the climb center for plumage color is displaced into the red species. So from yellow to red, which is consistent with our previous analysis. We find that patterns are similar for the second transect. So you can see here the points are also displaced from that red line into the flame-rumped genetic background. However, for the youngest transect, for our northern hybrid zone, we found that the climb is not yet displaced. So they're super concordant, which means that maybe there hasn't been enough time yet for the displacement to happen. And so why do we see a displaced climb? What's our interpretation of this displaced climbs? So many studies have linked displaced climbs to adaptive traits. So if this climb corresponds to something like plumage color, for example, there's a common assumption that the character is spreading because it confers some kind of reproductive advantage, like finding a suitable mate or increasing dominance against competitors. But testing these assumptions empirically is quite hard because it requires lengthy experiments in the field, and it's often difficult to test for females too. So evidence is often lacking. So building on those preliminary genetic data that I just told you about, we have a pattern where yellow plumage colors are moving from yellow into red. And we wanted to know the mechanisms behind it. So we have 3 non-exclusive hypotheses: that it was climate change and deforestation pushing the yellow birds up the mountain, or that they're larger population sizes in the yellow birds, so that's why they're contributing disproportionately. And finally, we have some evidence. So we have some evidence for the first 2, but for this project that I'm going to show you the results, we focused on testing what I mentioned just before, whether there was some kind of reproductive advantage of the yellow plumage color. So this is sort of sexual selection favoring the lemon rump males. So that means that either yellow males are more aggressive and then they are better competitors. And so they're winning when they're competing for females in the hybrid zone. Or maybe females prefer the yellow males, like they prefer the yellow plumage color for some reason. And so they're mating preferentially with yellow males. So female preference, very difficult to test. So we focus on testing if the lemon rump males were indeed more aggressive. And this is something that I had seen just from catching the birds in the mist nets. The lemon rump birds respond really well to the playback. They're really, really angry birds, whereas the flame rumps were much harder to catch. So to test for a relationship between aggressiveness and plumage color, we did some behavioral experiments to quantify these aggressive responses to taxidermic mounts that varied in plumage color. So we had 4 treatments, 2 mounts per treatment of lemon rump males, hybrids, flame rump males, and an ecological competitor, the blue-gray tanager. This was our control. And here you can see the variation of the individuals that we were able to collect. And so for the control, we chose the blue-gray tanager because it's sympatric for the whole range. So it inhabits the same areas of both subspecies. It's associated with similar habitats and resources. It's also a tanager. And so it was a good ecological competitor. And this would control for our testing of whether the males were responding to the playback or to the mount, right? So we wanted to see if the visual signal was triggering the aggression. So this is how realistic the mounts look in a natural setting. And so the reason why we needed real taxidermic mounts is because plumage color in these birds is very complex. And the color variation is not only based on pigments, but also on micro and nanostructures. And the yellow plumage has a peak on the UV. So I did try to do some 3D printing of mounts because I didn't want to collect. But for the life of me, I couldn't get the color to reflect what the real birds have, right? Like I just can't replicate that plumage color and that peak in the UV. So that's why we had to do some collecting to accurately test what was happening with these visual signals. So the experiments consisted of finding a singing male. I'm presenting a mount with any of the 4 treatments with a speaker hidden in the bottom. And so we kept the song conspecific, so the same song of whoever we were testing, because we were only interested in the visual signal. So we didn't want the song to affect in any way. And then we recorded everything that happened for the next 10 minutes after the playback started. And so there's 14 variables that you normally document. That is like how close the bird flies, if they attack, if they sing, if they perch in the mount. 14 different variables that are related with aggression. And so, for example, in this case, if the male was yellow, then we played a yellow song with any of the treatments we presented to each male. Same for red birds, so red song with any of the 4 treatments. And then same for the hybrids. Okay. And so we did the experiments mostly around the middle and the southern transects. We did 20 experiments to lemon rump males outside of the hybrid zone, and then 28 to lemon rump birds inside of the hybrid zone, 28 to phenotypic hybrids, 28 to flame rump males in the hybrid zone, and then 20 to flame rump males outside of the hybrid zone, which I'm going to call the allopatric zone, that zone outside of the hybrid zone. And this was just because we wanted to test if the behavior changes when they are in the presence of the other species versus when they don't interact directly, right? So when they don't find each other. Okay, so the million-dollar question, did it work? It worked really well. This video is slowed down for illustrative purposes, but they were very angry birds, and they really didn't like the mount. And this one is a double speed because it was very painful to watch the full length of the male almost destroying the mount. They were very, very angry. So we have quite a bit of different sort of behavioral patterns that we were able to observe. They like maybe just like chipped the mount a couple of times, just like bite it a little bit, or they would spend, this is how they sing, and they would like spend a long time just singing and displaying their back feathers. Also, they don't care about the cars or heavy trucks that go by the road, which is kind of wild, but that was really good to me because they were easy to find. Then there were other cases where the males wouldn't attack, but they would do this like dominance display where they would sort of eat in front of the mount, kind of like, this is my tree, you can't be here. And then there were other cases in which they would just like display, not attack, but be really close to the mount and display their rump feathers, just sort of like showing this very aggressive dominant signal. So when we look, oops, these are simple plots of presence, absence of attack. So attack, yes or no, during the experiments. So the number of experiments is in the y-axis, and then the amount is in the x-axis. And then we found that outside of the hybrid zone, the flame-rumped males are not very aggressive to competitors. So they only attack their same species and never a lemon-rumped or a hybrid mount. In contrast, the lemon-rumped males attack their own conspecifics more often and other mounts as well. So at least in this discrete way of looking at aggression, the lemon-rumpts are indeed more aggressive outside of the hybrid zone. When we look at the hybrid zone, the story changes a little bit. So flame-rumped birds showed higher aggression towards heterospecifics, so not their own species, and they attack lemon-rumped mounts even more than their own conspecific flame-rumped mounts. However, the lemon-rumped birds were extremely aggressive whenever they were tested with their same lemon rump. They attacked 100% of the times, and then they attacked the hybrids and the flame rumps in similar proportions. So in the hybrid zone, it seems that both subspecies are more aggressive to all treatments, which is expected because they're likely competing for resources, right? And then for hybrids, it seems that they're more aggressive towards either of the parentals than towards the hybrids, which is very intriguing. And then our control worked really well because none of them attacked the control. So it means that physical aggression was triggered by the mount. And then lastly, we had this, during the initial experiments, we noticed that the birds have this very conspicuous behavior that we called the fluff, which if you saw in the past videos, if you look at the male in the bottom corner, they would emit this very aggressive calls, and they would like open and fan like their tail and bow to the mount. Just to show this like very aggressive display of the rump, like the rump was their danger signal. And so this behavior was super frequent in almost all of our experiments. So besides the normal variables that I told you studies record, we also recorded how many times each individual did one of these fluffs. And so when we look at this aggressive behaviors, so when we plot the number of these fluffs versus the number of attacks, we can get a little bit more resolution on this data. So outside of the hybrid zone, red males don't attack as much. However, they display a lot. So they display a lot more than what they attack. Conversely, the lemon rump males display way less, but they attack a lot, especially towards their own conspecific. In the hybrid zone, we see the same trend, but both behaviors are intensified. So red males will display more than yellow males, and lemon males will attack more than flame males. And again, the number of attacks against lemon rump birds is higher in both. So the hybrids are doing their thing in the middle, but their behavior never reached the extremes of the pure parentals. So if we look at these results in the lens of the clients that I showed you at the beginning, we think that maybe the flame rump birds exert dominance, not with physical contact, but more with displaying their colorful, flashy signal, whereas the lemon rump males have reverted to using sort of like a simpler signal, and they resort to physical aggression to maintain dominance. So both behaviors are intensified in sympatry. So in the hybrid zone, they're much more aggressive. And because lemon rump males are superior competitors, they both receive and give more aggression, kind of. And then hybrids, which potentially have mismatched behaviors, either display too much in competition with lemon birds or attack too much in competition with flame rump birds. And so they may have lower fitness because of that. So with that, I hope I provided some evidence to support our hypotheses that in this system, male-male competition is probably driving this asymmetric flow from yellow to red. And so now we know the direction of gene flow, and we have a potential explanation of why it's going in that direction with the yellow males being superior competitors. We know that given time, this pattern likely repeats in each of our replicated hybrid zones. And so it seems that interbreeding is very common Right now, we don't have any evidence that it's being stopped by any chance. Like hybrids don't seem to be that unfit to stop hybridization. So for the future directions of my project, we're exploring some genes to actually find what are the genes responsible for plumage color and for the structural changes of the feathers. But for the sake of time and hopefully for some questions, I'm not going to talk about that part of my project, but if you're interested, I'm happy to talk to you about it and answer any questions. So to conclude, hopefully when we investigate these genetic mechanisms, we will be able to understand if the current evolutionary trajectory that they are in, which is they're sort of fusing, interbreeding, is not prevented by any means. Hybrids seem to be doing fine, and the hybrid zones are becoming wider. So will they fuse? Will they collapse? We don't know. Will we have 1,000 year just orange birds in both the Cauca Valley and the Pacific? We also don't know. But if they continue in this evolutionary trajectory that they are, it's very likely that that sort of orange phenotype is going to spread, or maybe the yellow will just invade and take over. So none of this would have been possible again with my field assistants. Elsie is going to give you a talk in a month. She's the postdoc in my lab. She works very far from Colombia in the Solomon Islands, but she went with me to help me do some of these experiments. And lastly, a very, very important person in Colombia who taxidermized the mounts for us. Very talented person. These mounts looked very real, and it's a really hard thing to do. So big shout out to Jorge. He's a professor at Universidad del Valle in Cali, in Colombia. You should all go there. Amazing birding spot. And I have an acknowledgement slide. A lot of ornithological societies have supported my work. And then I'll be happy to take some questions. I don't know if we have time. No time for questions.
Any questions? We'd like to record them, but to capture them. Wow.
In the United States, we have golden wing warbler and blue wing warbler. And if you go back data for New York State, you can see the hybrid zone has moved from the southern part of the state all the way to the northern part of the state. So do you have any thoughts on the hybridization of these two species? Are you aware of the golden wing, blue wing controversial?
Yeah. I've read some of the literature, and I think people have done some experiments to test like some of these aggressive responses, like whether one of them is a superior competitor, and that might be driving that movement. There's also like hypotheses on like whether females are preferring one of the plumages or not. But it seems that in that case, I think the hybrid zone has moved, but it's still narrow. So it means that contrary to what we see with the flame rumps, it's not that like hybridization is expanding. It's more than that that narrow hybrid zone is moving. Probably they also track changes in the landscape. So I think there's many different things that can influence hybrid zone movement. It can also be demographic changes in the populations and whatnot. Like if one population suffers a crash, then they have less individuals to contribute to the hybrid zone. And so the other sort of like gains some advantage, and then the hybrid zone can move. So there's like a few different aspects to consider. But I believe that's a case where they're probably not in this evolutionary trajectory where there's just like the gene flow is so pervasive in the hybrid zone. I don't know if they can detect hybrids that well either. Like some hybrids are genetically hybrids, but you can't really tell from their plumage that well sometimes. So it's tricky.
Thank you.
Yeah, of course.
I was curious whether you had a chance to go back to samples from the 19th century, for example, to see how quickly hybridization is happening or not happening.
Yeah. I didn't do it, but there was a paper on this same system that actually compared 3 different time periods in the southern hybrid zone. So they had Chapman expedition at the beginning of the 19th century, and then they had another in the 1950s, and then they went back and sampled it in 2010. And they were able to see that the hybrid zone was moving sort of like with this same pattern, so from yellow into red, but it moved, I believe, like 7 kilometers in 100 years. So it doesn't happen super quickly. But since this area in the mountains is so narrow, I believe 7 kilometers is a lot in that like very specific part of the mountain. So they did do that and found sort of a similar trend. And I actually used some of those samples for this study. So it's cool because that one is like a time frame comparison, right? And what I'm doing is more like a geographic replication. So this gives us a little bit more like a longer time frame to look at, right? Like we have like 6,000 years, 3,000 years, 1,000 years versus they have like what's happened in the last 100 years, which both are really cool. And so we have some data for that. Yeah.
Very nice talk.
Thank
Very,
you.
very, very nice. Just curious, the food sources for the carotenoids that apparently are getting converted into these colors, are they different in the different regions? And then, then that, of course, then begs the question, how do you sort of take that piece of the puzzle out of the equation as you're trying to figure out the, the hybridization or the color differentiation?
Yeah. So potentially their food sources are different, like there are different plant species, right? But it seems that the dietary carotenoids that they obtain from the different fruits are the same. So I actually have, I think I have a slide, maybe not. Yes. This is kind of hard to understand, but here we have like the two males, Ramphocelus, and basically I'm just showing you this because it's like they have the same dietary carotenoids in the feathers. So it's like they may eat, I don't know, I'm gonna say something. One fruit, I don't know which one. I know the scientific name, but let's say they eat different fruits, but they have the same pigments circulating in the blood at least. So we don't think that a difference in diet would drive at least that change like yellow and red. But I don't know if you're familiar with how carotenoids are metabolized, but the literature is like the state of the literature is like they eat yellow dietary carotenoids. That's how they eat most of their carotenoids. And they have this gene that converts them into red, right? So what we're exploring is what happened with that gene in the yellow subspecies in our case, right? It's like they don't have that ability or they turned it off, that path, that metabolic pathway of converting the yellow into the red, and they're only depositing the yellow ones. So that it's like it can be a very simple on and off mechanism, like they turn on and off that one gene. But so that's what we're exploring, like what's the genetics of that conversion.
And then for the orange feathers, which are some combination, you're saying, well, it's not like a light switch on or off. There's some sort of weakly expressed trait that might give rise to that.
It's so I'm actually writing a grant to do a postdoc on this because it seems that the color is determined by 3 different things. So you have the pigments, and then you have the microstructures of the feathers, and then you have the pigment concentration. So it seems that you can get orange either by mixing yellow and red pigments or by lowering the concentration of the red pigments.
Right.
So we don't know if they're not producing enough red pigments or if they're just mixing paints. And so we really, but for this, you have to like take the feathers and extract the carotenoids and do like HPLC analysis to see like how much they have of each pigment and which pigments they have. And there's so many carotenoids that it's a full-blown other world that I'm ready to tackle, but not in my PhD.
All right.
But it's fascinating. And then the one last thing I would like to say about that is that you have the pigments that determine the hue, right? So like whether you're red, orange, or yellow. Then the microstructures of the feathers, it seems that in males, they modify how light is reflected. So males have these microstructures that basically make them look more saturated, and females don't have them. So they're tricking the females. So they have the exact same pigment concentration, the exact same pigment quantity that females, but then they change the angle in which the barbs of the feathers are oriented. And so they reflect more light and made it seem like, look, I'm more shiny, I'm more brilliant, whatever. But then it's nothing metabolic or anything like that. It's just like they're amplifying that signal through a different way. So they're sort of cheating the system because it's supposed to be like a signal that they use to advertise quality. But then if it's not like costing them anything, is it really an honest signal? So yeah, it's a fascinating and so we want to map the genetics of this microstructure too, like just to see if they are different between the subspecies or different between males and females, or if it's just a giant network of carotenoids microstructures.
Sounds like we're going to have you back.
Yeah. Yeah. Yep. But
Did you look at population size and any impact this might have on your study?
yeah, so I actually did, and I didn't want to show it because it's kind of long, but we did look at population sizes across time. So we did like, how did the population size change in the past 100,000 years? Both populations are very similar. So both red and yellow, they're really close geographically, right? They're just separated by this mountain. So we expect them to suffer through the same sort of like climatic events and whatnot. And we found this like really huge crash in both, like around 10,000 years ago. And it coincides with a lot of reports of like human population crashes around the same area and other species. So it seems that there were some like climatic stability situation going on in that northern part of South America that caused this crash in a lot of different taxa. And they have been sort of recovering. So it was like, what we're thinking happened is like, they suffered this crash and their populations were sort of like really isolated. And then as they've been recovering and expanding, that's when they are coming into contact back again. We do see that the current population size for the yellow ones is higher because they're just like so much more widely distributed. So they have a higher population size, and that does like, that's another sort of hypothesis that we have, that definitely that higher population size can just by simple, there are more yellow birds to mate with kind of thing in the hybrid zone, that can affect that movement too. Like females maybe are not finding enough red males to mate with, right? So maybe it's not competition or female preference, maybe it's just there's nothing else kind of thing. So that's definitely another thing that we should that we need to consider, and we sort of like correct for it a little bit, but it's hard to know, I guess.
Anybody else?
Thank you. Thank you for having me.